Thank you so much, Professor Levchenko, for being with us.
Thanks a lot, Dora, for inviting me.
So systems biology is a relatively new field, and I'm wondering if you could tell us a little bit about what science used to be like before systems biology and how systems biology has sort of evolved science.
Yes, so science has been primarily dominated by the concept of reductionism, where we try to, in biology, but in other fields as well, we try to see if we can break down the system to some fundamental small elements or parts, study them separately from each other, and try to reconstruct how the system works. And it has done a lot of very important things, has brought about a lot of really important notions, but we believe that in the end, we cannot truly understand the function of the parts even, let alone the whole system, by looking at the parts separately. We believe that systems need to be looked at as systems, reconstituted, but also brought back to life in some sense through feedbacks, interactions, and synergies.
And so what are some of the novel insights or new discoveries that this approach of systems biology has allowed scientists to make or discover?
Yeah, so I think the notion of the interacting parts is very important if you try to understand how certain drugs, for example, work. Again, based on reductionist point of view, you can say, well, here's a mutation, here's something that I can do to try to fix it, either through genetics or through some chemicals. The problem is that in a system, even if something is broken, or if you really try to just look at a single part of the system, you may miss truly important interactions with other parts, and the system frequently reorganizes itself after something is broken. So when you try to fix it, frequently it doesn't work. It doesn't certainly work with many of the drugs we're trying to develop. So our insights very recently have been a lot about how these drugs may work, and more importantly, how they don't work, and how we can think about it and find frequently that it's all about these feedbacks that I mentioned, these interactions between parts. And so first we understand a little bit better, and more importantly, we find new ways to get around this problem, and frequently by looking at many parts, developing combinatorial interventions.
So it's a really exciting time. Maybe you could share with us a bit about what got you into science and what gets you up every day to come to work.
Yeah, so I think it's a very important thing to think about every day: what gets you up and what brings you back to work, especially when sometimes it's very hard. I think for me it's a couple of things. One of which is the sense of discovery I always get when I'm realizing that we sometimes look at something that has not been either seen before or appreciated before. So we get to discover new things. We get to understand things in new ways. For me personally, it's a huge bolus of energy and excitement. But also, as I started my path in science, I actually has always been somehow close to the clinical environments. I did my PhD at the hospital. And again, every day I walk through rows of patients that are either outside or inside the hospital, and you see with your own eyes the dire need for both understanding what may go wrong in our bodies, but also fixing that. And so I think we're all motivated by this, but for me personally, it's also very, very strong motivation.
Absolutely. Well, thanks so much for being with us.
Thank you, Dora.